Gratitude. Right. Good evening, people. I think you can hear me clearly. Hello, Komada. Right. Today we had. Can you hear me clearly? Clearly. Yes. So we had to discuss today the paper number 9. Paper number 9. Yes, yes. That means you can hear me clearly, right? Yeah. So we had to discuss paper number 9. I have given you the paper as well as the marking scheme both. Find it, take them paper, take the paper and the marking scheme. Right, paper number 9 and marking scheme. So we'll discuss the multiple choice questions first. MCQs. Right, first question, people. First question. First question. First question, which of the following is correct combination regarding function of subcellular structures? Which of the following is correct combination regarding functions of subcellular structures? Nucleolus, synthesis of rRNA, kaharine. SCR, synthesis of glycoproteins. SCR, RER synthesized glycoprotein. Peroxisomes, conversion of fatty acids into sugars. Peroxisomes, conversion of fatty acids into sugars. That is done by glycosomes, right? Lysosomes, detoxification. No, that is done by SCR and peroxisomes. Golgi apparatus, exocytosis of residue materials. No, that's done by lysosome. So we have the answer now. First one is the answer. Question number 2, people select the correct statement about cell organelles. Correct statement regarding cell organelles. Correct statement about cell organelles. Synthesis of phospholipid is done by RER. Phospholipids done by RER. Harnid, both RER and SCR involving in phospholipid synthesis. Synthesis of hemicellulose for the plant cell wall is a function of Golgi apparatus. Synthesis of hemicellulose for plant cell wall is a function of Golgi apparatus. Correct. And then the basal body of flagella has 9+0 microtubule arrangement, correct? And then basal body of flagella has 9+0 microtubule arrangement. Microtubule arrangement. What is that statement, people? Cell wall synthesis of hemicellulose. Cellulose and non-cellulose substances such as pectin, hemicellulose. Shall we check that? Golgi apparatus, Golgi, cellulose. The pectin was there, cellulose, hemicellulose, and pectin. I'm not sure about that. It says manufacturing of cellulose and non-cellulose wall compounds such as pectin. Such as pectin, not hemicellulose. So then it's a wrong one. Then it's wrong one. And then basal body 9+0 microtubule arrangement. Peroxisome is single membrane bounded vesicle with oxidizing enzymes. Oxidizing correct, ne? Oxidizing correct. Starch granules are found in stroma of the chloroplast and matrix of mitochondria. Starch granules are found in the stroma of the chloroplast. Starch granules are found in stroma of the chloroplast and matrix of mitochondria. No, you don't see mitochondria with starch granules. That is wrong. So then we can go with the given answer A, C, D agreed. Question number 3, people. Found the tissue of the plant which store fat. Synthesis of found the tissue of the plant which store fat. Synthesis of glycoprotein. Exocytosis. So A is incorrect. Okay, the previous one. Synthesis of phospholipid is done by RER. The RER, SCR, RER, both involving in phospholipid synthesis. Why do you say it's wrong? RER involving in synthesizing of phospholipids. RER synthesize membrane phospholipids. SCR also involving in synthesizing lipids including oil, steroids and phospholipids. Why it's wrong? Why? What's the problem? Correct me? What is the argument? Huh? It says SCR can also synthesize phospholipid. Only only synthesis of phospholipid is done only by RER. Then wrong. Then I'm wrong. Then has to be people. Then it has to be what? C and D. Do you have C, D? Then we'll go for that. Ne? Then wrong. Only keyword varadine. C and D. Then we'll take third one as the answer. So I think I have to change then the market scheme answer. What is the question number 2? Then answer has to be 3. Answer has to be 3. Right. Question number 3. Functions of tissues of plants which store fat. Sorry, found in the tissues of plants which store fat. Synthesis of glycoprotein. Exocytosis. Participate in photorespiration. Organelles for the above function respectively. Found in tissues of plants which store fat. Tissues of plants which store fat. Glycosome. Synthesis of glycoprotein. Glycoprotein nang RER neda. Rough. Exocytosis nang lysosome. Lysosome. And participate in photorespiration, mitochondria. Fifth one neda. Third one, synthesis of found in tissues of plants which store fat. Fat will take glycosome, not smooth endoplasmic reticulum. Glycosome. Synthesis of glycoprotein, rough ER. Exocytosis, lysosome. Participate in photorespiration, mitochondria, chloroplast, peroxisomes, all 3. So do we have another combination? I have taken third one as the answer. Third one first neither. Glycosome, smooth endoplasmic reticulum. Glycoprotein, people, RER, right? Glycoprotein RER neda lamai. Not SCR ne. Glycoprotein RER. Right? It has to be glyoxysome, rough ER, lysosome, and we can take mitochondria. Glyoxysome, rough ER. No, first one wrong neither. First one wrong because you can't take smooth ER for that. Wrong. So I'll take fifth one as the answer. Fifth one as the answer. Do you agree with me? Send me a chat lamai. I'll change the chat setting. Everyone kiladana. I can't see your chats. I have like separately people click your chat. You chat it, send the chat to the main line, okay? Main line, right? So then I can see the chat. So we'll third one people also, then I'm changing the answer. Third one we'll go with fifth one. Third one 5, agree ne? How is it? Question number 4. Production of steroids. Production of steroids. Production of steroids. Production of cellulose and pectin. Detoxification of peroxides. Helps in digestion. Function of smooth endoplasmic reticulum and Golgi apparatus respectively. Production of steroids, A you can take, ne? Production of cellulose and pectin, no. That's Golgi. That you can take as Golgi, ne? A and B. Detoxification of peroxides. Can you take the detoxification of peroxide, peroxisomes, ne? Helps in digestion, that is lysosome, ne? So then A and B, third is better, ne? Fourth one, third, hurry. Question number 5. Question 5. Which of the following is, so why 2B becomes wrong? Okay, which one? Which one? Which one? Which one? Which one? So why 2B become 2B become wrong? 2B can be neither. Synthesis of hemicellulose for plant cell wall is function of Golgi apparatus. It says not hemicellulose. Is it mentioned hemicellulose? It says pectin, cellulose, non-cellulose, and pectin. Collective modifying manufacturing cellulose and non-cellulose cell wall components such as pectin. So it's not mentioned hemicellulose specifically. All right. So we have to go according to the resource name. Right? In questions, people, every time we don't go anything beyond. We exactly analyze these resource book statements. If it is there, we can take. Otherwise, we can't. How is it? All right. Then we move to question number. Then we move to question number 5. Question number 5. Which of the following is best combination of cell junctions, its locations and functions? Best combination regarding cell junctions, location and function. Tight junction, location, skin epithelium, function, allow signals. Neither. Allow signals wrong. Ne? Make ganna. Anchor junctions, animal embryo. Neither. Embryo and heart muscle cells, what we said, people? Gap junction. Gap junction, heart muscle. Gap junction, heart muscle, prevent leakage, no. Tight junction, muscle tissue, strong binding. Tight junction prevent leakage, neither. Gap junction, animal embryo, exchange of materials. That is correct. Right. So we'll go for the fifth one as the answer. Fifth one, Harini. Question number 6. Question number 6. Question number 6. Select the common features of organisms in both phylum Nematoda and phylum Annelida. Common features for organisms of both phylum Nematoda and Annelida. Exist in marine, fresh and wet terrestrial environments. Exist in marine, fresh and wet terrestrial environment. Phylum Nematoda and Annelida. Marine, fresh and wet terrestrial. Common feature, eh? Bodies possess distinct head and segmentation. Wrong, neither. Absence of respiratory and circulatory system. Respiratory and circulatory. Annelida has. Wrong. Presence of longitudinal and circular muscles in the body wall. Longitudinal and circular muscles. No. Nematoda has only longitudinal. Eh? Wrong. Triple blastic coelomate. No. So we have the answer. Sixth, first one. Harineda. Sixth, first. 7. Anchor. Another feature found in phylum which includes animals that possesses features such as coelomic, unsegmented body without head. Another feature found in phylum which include animals that possess features such as coelomic, unsegmented body without head. Coelomic, unsegmented body without a head. Coelomic, who's this guy? Coelomic, unsegmented. Coelom is present in Annelida, so it can't be unsegmented. Coelom is present, unsegmented body without a head. Kauda Kandameta, Kandameta, right? Unsegmented without a head keyword, the radial symmetric ones are not having head. We didn't discuss it on that day. Right? Clitellum you can't take. Unisexually with external fertilization. Make a hurry. Flame bulb, no. Bisexuality, no. Nidoblast, no. Second is the answer. 7, second. Question number 8. Which of the following feature is also found in phylum which contain animals having tentacles, radial symmetry, and most marine habitat? Who's this? Who is this? Cnidarians, ne? Lamai podak chatting in Cnidarians, ne? Found in phylum which contain animals having tentacles, radial symmetry, and most marine habitat. Radula, tube feet, pseudocoelom, mesoderm, gastrovascular cavity. Second is the answer. Question number 9. 8 second. Question number 9. Some features present in two animals label A and B as follows. Endoskeleton, closed circular system, coelom. Endoskeleton, closed circular system, coelom. Exoskeleton, open circular system, gills. The phyla to which A and B could belongs are respectively. Phyla to which A and B belongs. Endoskeleton, closed circulatory system, coelom. Either has to be chordata or echondamata. Exoskeleton, open circulation, gills. Arthropoda or mollusca. So chordata, nematoda, wrong. Chordata, echondamata, wrong. Annelida, no. Echondamata and mollusca, correct. Right. Question number 10. Question 10. Question number 9-4. Harini. Question number 10. Question number 10. Question number 10. Which of the following statement is correct regarding secondary growth of plants? Secondary growth of plants. It occurs in all dicot species but does not occur in monocot species. It occurs in all dicot species but does not occur in monocot species. Which of the following statement is correct regarding secondary growth of plants? It occurs in all dicot species but does not occur in monocot species. Correct regarding secondary growth of plants. Occurs in all dicot. No, neither. All dicot secondary growth does not happen. Some dicots, ne? First is wrong. Elongated initials in the vascular cambium are oriented perpendicular. Elongated initials in the vascular cambium are oriented perpendicular to the axis of the stem or root. Elongated initials in vascular cambium are oriented perpendicular to the axis of the stem or root. Elongated initials in the vascular cambium are oriented perpendicular. Neither. Not perpendicular, neither. Elongated ones are present along the length of the axis of the stem or the root. Wrong. The cocambium, now the perpendicular ones are involving in forming vascular rays. What do you call the elongated ones are involving in forming xylem and phloem. Elongated ones along the length of the stem. Perpendicular ones are the short ones. The cocambium arise in the outer layer of the cortex in stem. The cocambium arises in the outer layer of the cortex in the stem. Cocambium arises in the outer layer of the cortex in the stem. The cocambium arises in the outer layer of the cortex. Cocambium arise. Cocambium arise in the outer layer of the cortex in the stem. What is this, people? Cocambium arise in the outer layer of the cortex. Now, in the stem, cocambium comes from the outer layer of the cortex. That's correct. But in the root, it's coming from the pericycle. Secondary phloem is not main component of the bark. No, it's the main component of bark. Wrong. Xylem vessels are present in both hardwood and softwood. Xylem vessels are present in both hardwood and the softwood. Softwood what is that, people? Gymnosperm wood. And then hardwood is the angiosperm. So you don't see that in xylem vessel, in the softwood. So we have the answer 10-3. 10-3. Question number 11. Which of the following has no relationship with the secondary growth of dicot plants? Has no relationship with the secondary growth of the dicot plant. Happens due to the action of the lateral meristem. Happens due to the action of the lateral meristems. Secondary growth of the dicot plant happen due to the action of lateral meristems. Following has no relation with secondary growth. Happen due to the action of lateral meristems. Correct, ne? Cocambium produce thick and strong cover towards outside. All right. The cambium of the woody stem. Cambium of the woody stem is single layer of differentiated cells. Cambium of a woody stem is a single layer of differentiated cells. Single layer, correct. Vascular cambium is a continuous cylinder of cells, correct. Cocambium of the root arise from the outermost, wrong, neither. Not outermost. It's coming from the Pericycle, so that is wrong. So 11th one, people, we can agree with has no relationship. 11th one, the scheme, it's given as 3, ne? But third one, we'll check it once again, people. The cambium of the woody stem is a single layer of differentiated undifferentiated. Differentiated can't be no undifferentiated. So we'll check this once again. Second and the fifth. This is differentiated layer of differentiated. Wrong, ne? So we have to select the no relationship. Co-cambium of the root arises from the outermost layer of the pericycle. Outermost layer of the pericycle, correct? I just said the outermost layer, right, of the pericycle, correct. Then we have the answer. Answer is 3. Right. So that is why you have to read the question fully, carefully, right? So you have to do these questions, different questions, and then challenge yourself. And then people practice this. So, all right. Now, question number 12. Question number 12. Question number 12. Right. Question number 12. Which is absent? Which is absent in softwood? Softwood ke anne gymnosperm wood, ne? Xylem fibers present. Xylem vessels absent. Xylem tracheids present. Parenchyma present. Cells with lignified walls present. Second one is the answer. Question number 12, people, the diagram given below represents the cross-section of a stem with secondary growth. Several tissues have been labeled as A to H. Which of the following indicate the tissues of the bark? Bark means, people, all tissues outside the vascular cambium. So D, C, B, A. A, B, C, D in other words. A, B, C, D only. Outside the vascular cambium, A, B, C, D only. So I'm taking then third one for 13th, third. 14th, people. Which of the following statement is correct? Secondary growth take place in all gymnosperm. Correct. Secondary growth occur in all gymnosperms. Yes. The xylem layer that transports xylem sap in woody plants are softwood. The xylem layer that transports xylem sap in woody plant is the xylem sap in woody plant is the softwood. Sapwood, neither, not softwood. Soft is wrong. Sap. Ilagata. Adventitious roots in monocots develop from pericycle. 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 Adventitious roots. We said lateral roots, not adventitious roots, lateral roots. Lateral shoots in dicot plants develop from axillary bud. Lateral shoots, axillary bud. Correct. Axillary buds, lateral buds. Correct. And then xylem vessels in springwood consist of large lumens. Springwood, large lumen, correct. So ADE, 14th, ADE. Market scheme, I have marked it as all. We'll check it once again. Secondary growth take place in all gymnospermia plants. Correct. Secondary growth take place in all gymnosperms. Correct, Nilamai? The xylem layers that transport xylem sap in woody plants are softwood. No, that is the sapwood. Wrong. Adventitious roots in monocots develop from the pericycle. No. Adventitious roots are roots arising from people in a plant other than the roots. Neither. Any plant part, when the roots arise, people, they are adventitious roots. We said people lateral roots, not adventitious. Lateral shoots in the dicot plants develop from the axillary buds. Correct. But about the axillary buds, where did we find any statement regarding axillary buds? Plant beginning. Plant consists of where axillary bud. Apical shoot apex के बालूम नहीं. Axillary elongation of the shoot due to primary shoot apex, dome shape. Right. Anyway, people, I can't find a statement on that. Lateral shoots develop from axillary buds. That is correct. Xylem vessels in springwood consist only larger lumen. Springwood larger, or summerwood smaller lumen. So ADE better. So we'll go with the fourth one. I think you agree with me. If you disagree, please send me a message. Question number 15, people, incorrect statement regarding growth of stems or roots. No intercellular air spaces in Casparian strip. Casparian strip, no intercellular air spaces. Incorrect. Actually, rather than saying intercellular air spaces in Casparian strip, it's better if we say that, people, rather endodermal cells. Neither. No intercellular air spaces in Casparian strip. Incorrect statement. It's better if you say, people, endodermis. Cortex mainly store carbohydrate and transport water. Cortex mainly store carbohydrates and transport water and minerals towards the endodermis. Inotopericycle solid vascular tissue found. Inotopericycle solid vascular tissue found. Cocambium produce coc cells to the interior and to the exterior and to the interior. Wrong, ne? Cocambium is part of the periderm. Correct. So we got the answer now. Fourth one for 15th, fourth. Okay, ne? 15th, fourth. So that's the end of this MCQ discussion. That's the MCQ discussion, ne? Right, people? What are the sections that you want me to discuss in the next paper? What section should we evaluate in the next paper? Tell me, I'll prepare the questions for that. What do you think that you're weaker? So send your chats to the main, everyone. Ne? What do you think? What are the sections we should prepare questions on this coming Saturday? Paper arang Saturday, right? You bring the paper. How is it? Plant response, photosynthesis, and kingdom fungi. Palamu. Lamai main chat ekade vandu. Main chat ekade danda. Oh, now you have to take one section from Unit 2, one section from Unit 3, one section from Unit 4. Right? Animal tissues will evaluate after some time. Now you very recently you learned that. Passe gamu. Animal tissues passe gamu taamatin. Karavitra animal tissues. Right? We just did, we can do only 3 parts, 3 sections. Right? We'll take fungi. Fungi gum hari. Good. Fungi. I'm another. Fungi. It need not to be in order, right? Need not to be in order. Random better, but then we should not skip any section. Photosynthesis. We'll take photosynthesis. 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 Plant responses. Plant responses. Plant responses. I will take that to paper number 10. Ne? This is for paper number 10. Padang karanna. Paper 10. Paper 10. Right? I'm doing a tough job, people. Right? Making questions and marking schemes, discussing it with you all. Neither. Yeah, I had from morning 8 o'clock to 5 o'clock, I had a class. Today's the exam batch class. Now I'm back again here. Right? Tomorrow I had to be in Kurunegala the whole day. Right? 8 to 5. So tough neither. And then Saturday in your class. Sunday once again Kurunegala. So so I had to spend a lot of significant time duration. So I can fix this only on Friday. Right? Significant time I'm spending. So when people are not answering people, it's very unfair. Right? It's very unfair when people are not answering. Plant life cycles will take later. Atti danne plant life cycle ne. We'll take it. Right? Maybe next paper, right? Paper number 11. Hari. Plant responses nang tiyak loku part when you say plant responses, response to the light, response to gravity, response to hormones, response to stresses. Plant responses. Right. And you have taken all tough parts this time. Photosynthesis, also long part. Right. Photosynthesis. And fungi, superi sections. Fungi. So in 15th question, why question 15, fourth answer is incorrect. Fourth answer incorrect. 15th, fourth answer. Coke cambium produce coke cells to exterior and interior. Now, it does not produce coke cells to interior. It just produces cells, but it produces coke cells to exterior. Right? Not to interior. Right? It's not mentioned in the resource that it produces cells interior and exterior, both sides. Is it given? But sir, it is present in the resource book. Is it? No, it does not produce coke cells. It produces cells. It can't produce coke cells to interior. Right? It can't produce coke cells to interior. It produces cells to interior. Just the parenchyma cells. Right? So, if it is mentioned, then it's wrong in the resource. Commonly referred to as the bark, crack peel, produce new cell layer, the periderm. Further, the coke tissues function as a barrier. Each coke cambium tissue produce compresses layers of periderm. Coke cells coke cambium produce coke cells to exterior and to the interior. Coke cambium produces cells to the exterior. Coke cells to the exterior and to the interior. Cells added to the exterior becomes the coke. Cells added to the exterior become the coke. Coke cells produced to the exterior become the coke. Exterior cells only becoming the coke. Okay. That is the reason. Right. Exterior cells become coke killer. There is another statement. All right. Now let's go to the structured paper. People, structured one. Structured. I have given you the marking scheme. So we'll follow that. Right, first question. What is the resolution power of the microscope? Minimum distance between 2 points that can be distinguished as separate points. Right, 2 marks for that. State 2 differences of transmission and scanning electron microscopy used to study internal structure transmission, scanning to surface wave, transmission to see electrons passing through the specimen, and beam of electrons reflects from the surface. Specimen are stained with heavy metals, stained with gold. State difference between eukaryotic flagellum and the prokaryotic flagellum, simple and complex. 20 nanometers, 200. Lac microtubules, 9+2. Extracellular, intracellular. What are integral protein? Protein molecules that are embedded randomly in the plasma membrane, which contribute to the mosaic nature. And we'll take it like that. You know, integral proteins are embedded in one or both lipid layers. Embedded. This word is important, right? Give an example of protein in the membrane act as enzyme. Microvilli on epithelial lining of some plant, some parts of the gut. Ekalakunai. What are chromatin? Complex of DNA and protein. Then draw diagrams to show structure of ER. Draw diagram to show structure of ER. So you have to show smooth ER and you have to show people rough ER with this diagram, okay? State 3 functions of Golgi. Collective modifying packaging, manufacturing cellulose, non-cellulose components and producing lysosomes. State different functions of cohesion protein and kinetoco. Cohesion is now holding two sister chromatids, attaching sister chromatids, chromosome arms of sister chromatids. Kinetoco attach microtubules to the chromosome. State cell organelles involving in photorespiration, mitochondria and peroxisome. You can take chloroplast too, neither. Chloroplast also you can take, ne? Chloroplast too, right? Chloroplast you can mention too. Photosynthesis kila but you know it's participating in the photorespiration. Hari, ilangata, what is cytoskeleton supporting structure of the cell? Label following diagram. Label following diagram. What's the diagram? Chloroplast, neither. What is A? Outer membrane. B? Inner membrane. C? In B? Intermembrane space. C? Inner membrane. D? DNA. E? Granum. F? Free ribosomes. G? Stroma. H? Thylakoid. 8 marks for that. What is the role of centrioles in cell? Role of centrioles in cell to produce astan spindle cell division. State composition of cell sap. Composition of cell sap. Water. Ions like potassium and chloride. Water-soluble colored pigments like anthocyanin. State functions of lignin found in plant cell wall. Provide hard and rigid matrix giving cell an extra support. Part 2. 1. Complete the following dichotomous key using appropriate numbers and letters to distinguish animals in the diagram. Distinct cephalization present. What are these animals, people? Cut the list of animals lagi. There was a picture given, neither. A is a Nigerian, ne? B is a sea star. C is nematode worm. D is annelida. E is a mollusk. F is an arthropod. Like distinct cephalization present. So someone's mic on low. Oh. Please keep your mics turned off. Alright. Next one, we will complete the following dichotomous key using appropriate numbers and letters. Oops. How do you documents, whiteboard? Notes. Okay, I'll take paint and we'll do it on paint. Ne? I'll share this. So how do you draw that, people? Distinct cephalization present. So if you take these animals, if you take what are these animals? What is this? A jellyfish, ne? Jellyfish. And then sea star. Then I have nematode. Then I have people. Nematode. What I have next is annelid, earthworm, ne? Earthworm. Then I have earthworm, and then I have cuttlefish, squid, ne? Squid, and then finally scorpion. So I can classify them into two. How do you classify them? First one, cephalization, no cephalization, eh? Cephalization. What I have for the animals with cephalization? Cephalization present, distinct cephalization absent. Exoskeleton present, exoskeleton absent. We'll take cephalization, no distinct, ne? Distinct cephalization. So what are the animals with no distinct cephalization? Jellyfish. What else? Sea star. What else? Nematode, ne? Nematode. Was a cephalization present in earthworm, squid and scorpion. So this is first step A, ne? This is B. There was a exoskeleton present. Exoskeleton present, I think we can divide this one, ne? Exoskeleton present. Exoskeleton present. Exoskeleton. Here, no exoskeleton, ne? No exoskeleton. Exoskeleton present, I think, can take scorpion. No exoskeleton, earthworm, squid. Then next one is clitellum present, clitellum absent. That is number 3, ne? 3A. Clitellum. No clitellum. Clitellum present, earthworm. So 3A earthworm, ne? No clitellum, squid. Then this one we can classify. What is the step, people? Mouth underside. Mouth underside, mouth not underside. Mouth not underside. Mouth underside, you have jellyfish and sea star. Not underside, nematode. Then mouth underside, now what is this? Number 4, ne? Number 4, A and B. Then the last one, number 5. Tentacles present around mouth. Tentacles. That is 5A. No tentacles. Tentacles present, what do you have? Jellyfish. This one is sea star. Right. So based on this information, now you can fill. Ne? Based on this, you can fill. Right. So you can fill, and once you do that, then you get obtain this distinct cephalization. Okay, these animals have A, B, C, D. Oh, ne? A, B, C, D. Ne? Okay. So instead you put A, B, C, D. Hari. Right. I think this is okay with you. Then what is the animal group with first appearance of little complex nervous system? Platyhelminthes. State the unisexual invertebrate animal, nematoda, some annelids, most mollusks, arthropoda, chondromata. And then what are the body parts of mollusca? Muscular foot, visceral mass, and mantle. What is growth? Irreversible increase of dry mass associated with the development of an organism. Following is diagram showing a student show structure of the monocot root. Hari, state 3 points why this diagram is wrong. Xylem and the phloem should be located separately. Oh, there should not be cambium present between the xylem and the phloem. Neither. Xylem tip has to be turned towards the outside, not to inside. Ne? From where the lateral shoots develop from the dicot stem. From where the lateral, what is that? Lateral shoots develop from the dicot stem. Axillary buds. I think in your question paper, from where the lateral develop, the lateral shoot, that's a small weakness in the scheme. And then people state how dicot vascular bundles are different from the monocot vascular bundles. Dicot vascular bundle cambium present, no cambium. Vascular bundle is not surrounded by sclerenchyma, surrounded by sclerenchyma. Vascular bundles are arranged in a ring, scattered. What is secondary growth? increasing in the diameter of the stem and roots in plants due to new cells produced by lateral meristems. Give following events in order to placing number in front of the event. Would accumulate and lignified. Give following events in order by placing number in front of the event. Vascular cambium form, first event. Would accumulate and lignified. Co-cambium form. Lenticels form. Co-crack and peel off. Label bark, cork, heartwood, sapwood, and annual rings in the following diagram. Cork. This is the cork. The outermost layer is a cork. This is the cork. It was a heartwood. Heartwood is wood present at the middle. Heartwood. This is heartwood. More closer. Next one is the sapwood. Sapwood is closer to the vascular cambium. Closer to the vascular cambium is the sapwood. It was a annual rings in the diagram given below. Annual ring. How do you show annual ring? Yeah, dark and light band. Annual ring. Okay. So annual rings. I showed you that last week. So draw the annual rings. And then people state two differences between soft and the wood and the sapwood. Two differences between softwood and the sapwood. Softwood present only in gymnosperm. Sapwood present in both gymnosperm and in dicot plants. Xylem vessels are present in some sapwood, but xylem vessels are absent in softwood. So those are the differences. Right, moving to the next page, people. State the difference between growth ring and annual ring. Light and dark band seen in the cross section of the stem due to thick secondary xylem formed by wet season compared to the other seasons are growth rings. So when the favorable conditions present, due to favorable, unfavorable conditions, rings appear, they are growth rings. In temperate region, wood form in spring and wood form in the rest of the growing period called the summer wood. Those are collectively known as annual rings. So annual rings are the rings form over 1 year. They form over a year. Right, then we will, in the essay part, I have asked you to describe the structure and the function of the cell wall. Structure and the function of the cell wall. So this is coming from unit 2, plant form and function. Sorry, unit 2, cell structure and function part. Right. So cell wall is an extracellular structure and not only present in plants but present in fungi, protista, bacteria and all. Chemical composition is vary from plant part to part, species to species. Cell wall is made up of cellulose pectin. Plant cells generate two types of cells, primary and the secondary cell walls. Outermost part outside the cell wall is the middle lamella. Young cells secrete primary cell wall and just outside the primary cell wall, middle lamella. Middle lamella glues the adjacent cells together. Primary cell wall is permeable, thin, flexible, composed mainly of cellulose. May all live the lamai essay? How many of you wrote the essay? Water can move freely through in and out. Secondary cell was the Miguel Arangin. How much check karna me sati harida? Me sati nan check karna. Guti tamai carbonati at Harda Arangin the inisa, and it died a guti me sati. Right. In addition to cellulose, impermeable substances such as lignin, suberin also present. Lignin cement and anchor Cell wall protection and support allows development of turgidity and prevent bursting of the plant cells, right? Make the apoplastic pathway, maintaining cell shape, etc. Right? And then people, my second question is regarding the secondary growth. Describe the secondary growth of stem. Right? We discussed the theory part, how it happened, but I think you better draw a diagram also. Secondary vascular tissues produced by action of vascular cambium. Typical dicot stem vascular cambium consists of continuous cylinder of undifferentiated cells, single cell layer in thickness, right? Located outside the pit and the primary xylem, and inside of the cortex and the primary phloem. So where the cambium present vascular cambium. Then these cells are meristematic. They produce cells towards outside and inside. Secondary cell xylem to inside, secondary phloem to outside. In the cross section, vascular cambium appears as a ring. Some initials are elongated and produce xylem and phloem, and they produce tracheids phloem cells. Others are shorter and oriented perpendicular, producing parenchyma. Secondary growth continues over many years, would accumulate during early stages. Epidermis push outward, cause it to split, replaced by the cocambium. So coc cells are produced. Cocambium produce coc cells exterior. Cocambium and the tissues produce collectively called as the periderm. Right? Fully labeled correct diagram mark only. Right? Those are the points expected. හරිද? එහෙනම් ළමයි අරගෙන එන්න මතක් කරලා, right? When you come to the Saturday class, bring this paper that you marked, answered. I'm going to check that. Right? පොඩි time එකක් කරන් check කරන්න. Right? And we'll give a small treat for those people who are not writing the papers. පොඩි treat එකක් දෙන්නම් වෙනවා. Then thank you people for joining with me tonight, and I'm hoping to see you soon. Regular weekend class, right? So big thank, and then have a nice time. Have a nice time. So see you all people. Right, goodbye. Night. Good night.